Okay, once again, welcome everyone. Before we continue on to Tally's presentation, I want to give a quick overview of the IWGSC. We are an international nonprofit consortium with 3,450 general members, 943 institutions and companies, and located in 76 countries. And I want to take this time to acknowledge our sponsors that make today's webinar possible. So let's all thank Arbor Biosciences, BASF, CIMMYT, Florimont Desprez, INRAE, Kansas Wheat, and RAGT. So thank you to our sponsors. As most of you know, the IWGSC was instrumental in the production and release of the first bread wheat reference sequence that was released in 2017 and published in 2018. Our mission since that time is to develop additional tools and resources to enhance breeding through an increased understanding of the molecular basis of traits and their allelic diversity. And to this end, I'm very happy to announce our latest IWGSC project. It's the Wheat Diversity Project with the goal to sequence 12 landrace genomes that encompass worldwide diversity in wheat. And as well, we are updating the Chinese Spring RefSeq to platinum level quality. So among the other aims listed here, this was made possible with funding from the National Science Foundation, the European Research Council, and is an international collaboration between scientists at INRAE, University of Paris-Saclay, and University of Georgia. So I want to acknowledge the funding for this project, and you'll be hearing more about this project as it goes forward. So please stay tuned. And also, I'd like to say that our next webinar series, we're planning for sometime in January, hopefully, if not February. So stay tuned for that. And as well, I want you all to know about the upcoming workshops, particularly at PAG 2025 in San Diego. As usual, we'll have our main workshop on Saturday in the morning, so that'll be Saturday the 11th. And then a second workshop from structural to functional wheat genomics, and that'll take place Tuesday afternoon. And for further information about the IWGSC and the PAG platform, you can go to this link right here. So we look forward to seeing you all in San Diego for those who can make it. Okay, so before we get going, I want to introduce all of you to the dashboard. So this webinar will be recorded and it's going to be posted on the IWGSC YouTube channel. So the link is here. And for those who are following along, we'll have a presentation followed by question and answers. If you have a question, please use the Q&A button. Submit your questions. They will be saved for the end. We will try to do all of the questions that were asked. And given time restraints, if Tally is unable to answer the question because we run out of time, he's kindly offered to answer them in writing. In addition, please monitor the chat panel. If you have any kind of technical issue, you can use the chat panel to raise awareness to the technical issue, or if you want to receive communication from the person who's administering this webinar, Isabel, you will also find communications there as well as access to handouts and things like that. If you haven't already signed up to be a member of the IWGSC, I certainly welcome you to do that and register to our mailing list so that you can stay in touch with our community. Okay, so let's get to Tally Wright. Um, Once again, he's a group leader in the crop quantitative genetics at NIAB UK, and his talk is using applied quantitative genetics to uncover the potential of diversity-enriched wheat. So thank you very much, and thank you all for your participation.
I hope you can all see the slides and hear me okay. Is that a yes, Josh? Can you hear me okay?
Yes,
Yeah,
yes, I
thank
hear
you.
you.
Good, thank you.
Yeah.
Well, good afternoon from the UK, everybody, and thank you for coming to this webinar today. My name is Tally Wright, and I am a research leader in crop quantitative genetics within the plant genetics department at NIAB. And today I'm going to be talking about some research into our genetic diversity resources in wheat. I'm sure I don't need to tell this audience, but genetic diversity is paramount for making future yield potential improvements for food security going forward. And really captured in this quote here from Julia King in their recent plant genetic resources paper, there are a lot of understudied accessions of wheat in collections worldwide, just under 1 million, which may hold potential for yield improvement going forward. And that's the case at NIAB on a slightly smaller scale. So we have large collections of plant genetic resources. And this is really going back, so this has been collected over sort of 10 years of funded projects, typically by the BBSRC. So you can see some of them here. And this is looking at accessing genetic diversity reserves and going back to this hybridisation that happened in the wild 10,000 years ago between tetraploid donors, Triticum dicoides, and the Agellox tauschayi, maybe the less explored D-genome donor. So we have collections of plant genetic resources for Triticum dicoides on the AB side, Dicoccum, which is cultivated Emma, Triticum dicoides is wild Emma, Triticum durum, and some durum land races, over 300 of those. And then on the Agellox tauschayi side, we have 200 accessions, and then we've been forming synthetic hexaploid wheat from those, which I'll go on to talk about in a bit more detail. We also have some collections of elites as well. So these are our GWAS panels. We do a lot of comparing between the diversity available in these resources versus what's present in the elite gene pool. In terms of accessing the tetraploid diversity, so we have these tetraploid resources that I was just discussing. And here we make direct hybridization with modern wheat, hexaploid wheat, to form F1 hybrids. Then via backcrossing, we form a series of BC1 derived lines. And via backcrossing, we tend to sort out the ploidy issues between the tetraploid and the hexaploid. And these form our forward genetic resources for exploring that tetraploid diversity. And so that diversity, you can see in this picture here, there is some fantastic diversity available. And in this principal coordinate analysis, you can see that we've sampled lots of different population structure groups from those tetraploid donors. Then in terms of the Agellox tauschayi, so on the D-genome side, we have large collections spanning our large collection sort of spans the geographical range of the species. So we've colored the tauschayi collection sample dates here by lineage group. So you can see that we've got lineage 1, which is the one which is geographically separated by the Caspian Sea. Lineage 2, which is under the Caspian Sea in northern Iran. And in this principal coordinate analysis space, you can see we sampled different lineages of the species. And even we have an individual from lineage 3, which is the more recently discussed one. So Richard Horsnell, Bethany Love, and Bright Fiakiye are here, and that they were doing a GWAS study on this collection in the summer. But what I'm talking more about today is our work with primary synthetic hexaploid wheat. So I'm sure a lot of you are familiar with this concept here, but we've been forming primary synthetic hexaploid wheats using those diploid resources. So we take an Agellox tauschayi line, hybridize it with an emasculated durum, and that forms an F1 hybrid. Then via embryo rescue and chromosome doubling, we end up with the primary synthetic hexaploid wheat. So we do that using a different tail shy for each primary synthetic that we create, and we've done that 64 times at NIAB. Richard is the person doing that, shown in this cartoon here. So he's our magical synthetic hexaploid wheat machine. So this was done by Brandy Wolf's group in their sort of Nature Biotechnology paper that they recently released, looking at the diversity of some of the Aegilops tail shy. We also have 36 synthetic sourced from external sources as well. So this has been a large ongoing amount of work from Richard at NIAB. So as part of these projects, we have a lot of this diversity resources, which we use as parental genetic resources genotyped. So that's using arrays such as the 35K axiom SNP array, and also genotyped using Cas markers as well. With a lot of ongoing consortiums and collaborations, we're moving to get a lot of these sequenced as well. So we have 2 chromosome level assemblies via the tetraploid pangenome efforts, some Durham assemblies, again via the tetraploid pangenome efforts, and then 33 accession sequence as part of that Nature Biotechnology paper that I discussed in collaboration with Brandy Wolf's group. So we also have some of those, the tail shy side. We have some of the synthetic hexaploid wheat. Just to say those 33 accessions are the ones which we used, or some of the ones which are used in our synthetic hexaploid wheat formation. Then on the synthetic hexaploid side, we have 2 accession sequence of chromosome level assemblies in collaboration with the Welker Sammer Institute. And I'll talk briefly about 2 of our magic populations, but we have 8 accessions with chromosome level genome assemblies, which is the founders from the magic elite population. And then in terms of magic diverse, those have exome promoter capture sequences. So how do we capture these genetic plant genetic resources for forward genetics? We typically form multi-founder populations. We do this because they're high in statistical power, so they're great for genetic mapping, and they're high resolution. They involve a lot of recombination, so they're quite accurate in finding QTL. They also capture a wide amount of genetic diversity, and that's via using multiple founders, which is one of the clues in the name. So there's sort of 2 flavors here of the multi-founder populations that we create. I'll talk briefly about magic, but most of my presentation is focusing on our nested association mapping resources. So magic, you typically take 8 to 16 different founders, have 3 to 4 levels of intercrossing, and at that stage you then form recombinant inbred lines. Here you get a lot of recombination due to those amounts of intercrossing, and you end up with quite a precise mapping panel for QTL mapping. On the nested association mapping panel side, so this is what we do with most of our diversity resources. We take a donor and then backcross it to a common elite parent. So it's a series of basically around 50 nested populations, if we have 50 donors, all crossed, which are derived from all crossing a different diversity donor to an elite. And when I say elite, I'm normally talking about either a UK winter wheat elite, typically Robigus, or a UK spring wheat, a UK spring wheat paragon. So that's the spring background. I'll touch briefly on the magic population. So we have 2 populations which have been quite well studied at NIAB. This is the magic elite population, magic diverse, typically have around 500 to 1,000 genotypes in them. And at the time that they were created, via using elite wheats, and they captured a high proportion of the UK SNP polymorphisms at the time based on that elite selection. So the magic elites were based on a breeder selection. Magic diverse, I think, was selected to capture diversity. If we just look at the magic elite population in detail, you can see it's been quite well studied. There's several BBSRC projects that have looked at that population in detail, looking at things like yield components, photosynthesis, and these typically involve collaborations with breeders as well. There were also some PhD projects looking at disease resistance and some internal collaborations, all based around on this mapping population. So our work there has been quite extensive. The reason I wanted to touch on the magic populations is we have a third population called Magic Wilder. Unfortunately, the magic diverse name was already taken, so we went with Wilder here. And here we've made a series. Here we've taken forward as the founders of the population a series of BC1 derived rills. So They are ultimately in the paragon background, so they're spring elite wheats, but they contain 25% of the genomes are introgressions from synthetic hexaploid wheat, Triticum dicoides, Triticum dicoccum, and Triticum durum. So a lot of wild diversity or resynthesized diversity is present in this population. These are the founders here. It'd be a little bit more interesting if I'd captured them at a later stage, but this is what they currently look like in the glass house. And after 3 rounds of intercrossing, we have 1,000 reels currently at the F5 generation. So a lot of work has gone on there to make that population, and it's currently ready to be genotyped and phenotyped to assess the variation, similar to hopefully what we've done with the elite wheat magic populations. So this is the side of the work that I'll be talking about in more detail here. So we have nested association mapping panels, and these again combine high statistical power with large diversity capture using around 50 different diversity donors in the population. And what we're typically doing is taking our synthetic diversity and our tetraploid diversity and backcrossing it into those 2 UK elite backgrounds that I spoke about, one winter and one spring. We do that because we want to make a useful population from the academic side for genetic mapping, but also from the breeders side. So we want breeders to come and take forward this material for use in their own pre-breeding programs. So making it more adapted to a UK environment is quite key. This is what the NAM populations typically look like in the principal coordinate analysis space. So this is made using curly-wurly. So we have our diversity donors over here, our recurrent elite over here, and the nested population sitting closer to the elite, as we'd expect for back-cross-derived lines. So these 4 NAM populations were made as part of the Designing Future Wheat project funded by the BBSRC. So we have this one that I'll be talking about in most detail today. So 54 biparental populations, over 3,000 recombinant inbred lines, 54 synthetic donors that each went into forming those populations, and that was via a back cross to a winter recurrent elite, which was Robicus. And each of those 54 synthetic donors each have a unique Taoiseach as their parent, or mostly have a unique Taoiseach as their parent. So we have the parallel population with a spring recurrent elite, and then 2 more tetraploids derived NAM. So again, similar to the synthetic side, but this time using dicoides, dicoccum, and durum donors as the diversity donors. We have another synthetic NAM that was formed as part of the WISH panel. And then we have another synthetic NAM, which is what we call the WISH panel. So this is formed using SIMET elites in an earlier project. And then the magic wilder population, which is the magic population I discussed, and chromosome segment substitution lines that I'll go on to discuss in more detail. So this is an overview of some of our diversity resources at NIAB that I wanted to show. So this is our work with the winter wheat, synthetic hexaploid wheat NAM. So this came out in Theoretical Applied Genetics this year. So please go and have a look at that paper if you want to read more about it. And this is our sort of workflow for creating the NAM. So I've already spoken about synthetic hexaploid wheat formation. So I'll skip to this step here. So we have 54 of those primary synthetics. Most of them are formed using unique Taoiseach. We then backcross to Robicus here, the winter UK elite, and then from the BC1F1 we take several streams forward to form a population of around 60 individuals which are BC1 derived, go for a single seed descent program, get to the BC1F4, bulk the plants up in the glasshouse, and then sow them into a nursery field trial, as you can see here. These trials we called our core nurseries, and breeders, as part of the Designing Future Wheat program, would come and make selections on this material, and they were free to take it forward for their own commercial programs. And we'd also take some of our own measurements. So we'd look at flowering time, plant height, and yellow rust resistance in these trials. Here you can see the winter wheat Robicus. You can see a primary synthetic wheat ear here and a synthetic derivative. So you can see it's a much nicer looking wheat than the primary synthetic, and a lot more breeder friendly, but also still containing a lot of novel diversity. So this is some of the population structure of the population. So this was completed by Bethany Love, who's a postdoc working in the genetics department. So here we have a similar pattern to what we observed before, our primary synthetics, the population lines, and our recurrent elite up here. If we just look at the D genome of those primary synthetics, which is what we're doing here, we can see that they go back to the original lineages of Agelots tauschayi. So we have lineage 2 here with the Robicus D genome sitting in there as we'd expect, as that's where the original D genome donor comes from. We have that lineage 3, the more recently discussed lineage that originates from modern day Georgia, and lineage 1. So it's much harder to sample or form primary synthetics from lineage 1. So this group is typically underrepresented. So I've gone through those numbers already, but just to say, and just to say, you can see the geographical range of the species again there, and we formed 47 of the synthetics. We had 47 unique Agelot tauschayis and 15 different tetraploid donors, and most of them were made using, or most of them included in the population with NIAB synthetics, but there were some from external sources as well. So this is the genetic analysis workflow, and with over 3,000 individuals, this was a lot of work and quite challenging. But what we did here, it took several years as part of the Designing Future Wheat program. So we extracted DNA from BC1F5 individuals, sent that DNA to the University of Bristol, where it was genotyped on the 35K SNP array. We got that genotyping data back at NIAB, and then using Axiom Analysis Suite, we did a lot of manual SNP calling and marker quality control, and a lot of individual quality control of over 3,000 individuals to try and get accurate data. We imputed missing data with random forest, and then anchored markers to the physical reference genome using LD bins. Prune markers on linkage disequilibrium to remove duplicates, and also when we're working with the arrays, we find that a lot of the markers aren't that evenly distributed. So we typically thin the markers out for estimating kinship, and that gives us better correction levels in terms of association mapping models. So that's what we did there, and then this goes into an association mapping pipeline. This data is currently hosted on our GitHub account, so you can view the data there and some phenotype information about the population. So I mentioned that genotyping that many individuals was challenging. So phenotyping them is also a little bit of a nightmare. We had sort of 5 core nurseries as the population was staggered. So over a series of 5 years, different nested populations went out into the field for assessment by us and for selection by breeders. In 2021, we managed to secure some funds to grow what we called the Mega NAM nursery. So this was the whole NAM population with over 3,000 individuals grown in a single field trial. It was, challenging. It took 2 days to walk it to score for a single trait, such as orm presence. It was a lot of work, but I think it paid off. And here we can look at some of the phenotype data from those 6 trials. So we got flowering time, plant height, and yellow rust infection. Yellow rust infection was scored in the first 2 core nurseries, which were actually untreated nurseries. We started then treating the nurseries normally with fungicides. We were worried that it was kind of ruining the physiology for the other traits. In the 2021 mega trial, we managed to score everything by scoring before a fungicide application to get a yellow rust infection score. And you can see an aerial image of the size of the trial there from NIAB's data science department. So as I said, the wheat breeders have been selecting this material over the Designing Future Wheat program. And those are the companies that came and made selections. And it's often hard to find out how far a genotype makes it in their commercialization program or their commercial program. But we had some positive feedback from Matt Curtin from DSV. So Matt let us know that a NIAB synthetic derived line had made it as far as national listing in his commercial trial. And although it didn't make it as a variety, it was still named Gandalf. And Matt said he called it that because it was bearded and weird, which really does reflect some of the diversity we observe. It's a great resource, but hopefully this line will go back into population improvement. And that's a picture of Gandalf there, not Matt. So onto the selection side. So we have looked at the selection data, so in terms of what genotypes have been selected by breeders over the program. So if a genotype was selected by any breeder, it's marked as orange there, and a square represents a synthetic derivative in the program, and blue is something that wasn't selected. So if we look at this selection pattern, so this is the tetraploid background, so it's reflecting the original tetraploid that was used in the primary synthetic that made that synthetic derivative. And you can see that although it wasn't properly randomized as a selection experiment, you can see that there are some visual trends. And this HO501 background is the durum, which was commonly used for NIAB synthetic wheat creation. And actually, when you look at some of the rarer durum backgrounds, they had a much higher selection rate than HO501. In terms of the Taoshai lineages, so going back to the pedigree of the actual Taoshai, you can see lineage 2 was more favored and was the most common as well. So with all this genetic mapping data and phenotype data, we then completed a GWAS analysis. So here we have trait data for flowering time, plant height, and yellow rust resistance across those 6 different trials. 26 QTL were mapped in total. Here I'm showing the data for just yellow rust from those 3 trials that we scored in. So there's a really nice hit on 4D, and it's the same peak marker each time. And here we increase individual numbers, so that's why it gets more significant. The peak marker from that QTL at the start of 4D, so it blasts against the reference sequence. So 400 kb away from a known rust resistance gene for yellow rust, which is commonly found in Taoshai and primary synthetics. But it was exciting because it's not very common in many land races or cultivars. And until this point, there was no evidence of it working in a UK wheat background or a UK environment across 3 years. So it's shown that it's effective here, which is a really nice example of capturing some of that diversity from the Taoshai background and seeing it work in a hexaploid background. Yeah, so 37 out of the 54 synthetics carried the resistance gene. And then if we looked at what was being selected by the breeders, we could see that it was actually enriched for that resistance gene as well. So the material being taken was typically enriched for the resistance allele at the peak marker. So it'd be nice here if I could flow into the resistance in the chromosome segment substitution lines, but one of the only synthetics that didn't carry the resistance gene was used as the CSSL donor years ago without us knowing. So that's unlucky, but you can still look at other trait variation here. So what we ultimately have here is chromosome segment substitution lines, and this is ultimately paragon, so a series of paragon lines with small introgressions from either a primary synthetic wheat, and because of that, it has introgressions from Ajox, Taoshai, and durum as well, or TD140, which is the AB side, the orange introgressions here, which is from a triticum diacoles line. So we ran a series of marker-assisted backcrossing here to get to the BC4F4, and then genotyped on the folio 5K, and at that point made a selection over which line to take forward. So we were trying to select for small introgressions into the paragon genome to have a kind of tiled effect of these introgressions from the wild across the paragon genome for the AB on the TD140 side, and on the D-genome side from the primary synthetic. So this work is published in the plant genome, so you can take a look at the paper there. And the idea here and why I wanted to mention it is we can look at our trait assessment in those NAM populations. In this case, we're looking at leaf glaucosity or waxiness. So the most common phenotype is the waxy or glaucous leaf, and then we typically have some of these waxless types coming from the wild background. We can do some QTL mapping, find that there's a peak marker on 2D in this case that we want to look at, then go to the CSSL lines and look to see if we have an introgression from Agelops Taoshai within that region, which we do for 2 of the CSSLs shown here. We can then see if these share the same phenotype, which they do. These are the only 2 CSSLs which are or D-genome CSSLs which are non-glaucous or waxless, and then conclude that we know that the allele linked to this is within, or the gene expressing this is within this introgression region, which is a 20 megabase pair introgression region. So this is a nice resource for then doing some fine mapping and ultimately comparing this genotype here with paragon. You've got a nil for your QTL and trait of interest. So the other side of my work with the synthetics I haven't spoken about in much detail here, and more from the breeding angle, the breeding interest is their yield performance. So as a pre-breeding resource, that they're quite attractive because we often have a lot of individuals which are outperforming their recurrent elite. So in this case, we have Robigus, and this is a trial from Rothamsted in the 2022 academic toolkit. So you can see that there's a lot of synthetic derivatives which outperform the recurrent parent, and even in some case that they yield as high as more recent elites. And the fact that they have this high yield potential and a high yield performance, and we know that they have a lot of these new introgressions across the D genome, as you can see with this plot made by selection tools. And these are some synthetic derivatives that make them desirable candidates for pre-breeding programs. And to emphasize that, I've been working closely with Dr. Karis John Bajaya over the last couple of years, who was making selections. So Karis is the pre-breeder from KWS UK. Karis has made selections from those Designing Future Wheat programs, material that he wants to test at KWS, and he took 406 lines forward that we have phenotype and genotype data for. Assessed them in yield trials over 4 years, and we found that when we do association mapping for this, we get no QTL, which this is for grain yield, and that's not uncommon. There's a lot of diversity present in the NAM population, and because it's a backcross population, that diversity is not always represented as in as high levels as you'd like, because ultimately you're starting off with only 20%, 25% of the diversity coming from the wild lines. So that's what we find with grain yield, with a trait which is controlled by many genes and also is affected by the environment. So we were interested in looking at more of a genomic selection-based approach here. We took those 406 lines as the training population, and then we can hide some of the data, which is what we call cross-validation, see how good the prediction is using the GBLUP method. And when we're happy with the prediction, we use the whole training population to estimate our prediction. We then use genomic prediction, such as GBLUP, and then use our test population where we just have genotype data, but no phenotype data, to make a prediction on how good the lines are with just genotype data. So this is helpful if you can't always do phenotyping of everything. Like in this case, we have a lot of material and we can't phenotype everything as it's too expensive. We have bulk seed available for 15 families, so we use those going forward to make our predictions. And our predictions over the highest yielding and the worst yielding went into what we call the independent evaluation trials. These are 2 replicated trials grown at KWS this summer, where we predicted the worst versus the best to see if the prediction's working. This is the prediction accuracy in terms of the cross-validation. So here we've run the prediction around 1,000 times, each time hiding a proportion of that training population, is like we call the cross-validation set, and each time selecting a new set for the cross-validation. And what's interesting to see is that the prediction is pretty good. It's around 0.5, and that's typically what we'd expect for a genomic selection for yield. It's okay. But what we find is that depending on what ends up in the cross-validation set, the prediction is quite unstable. Sometimes it's very good, sometimes it's not very good. And I think that's when we look at what was sampled in the training population across the years, we can see that some nested populations, which is what we're ordered on here, were actually very well represented, where in some cases we didn't have as good representation. So if the genetics aren't in the training population, it's very difficult to make an accurate prediction. When we look at the predicted families from those 15 nested populations, like nested population rather than family, we have quite interesting results in terms of then going back and comparing to what the breeders were selecting for in the Designing Future Wheat program. So the 2 lowest yielding populations, one was the lineage-free donor, which is interesting, and also in that Ho501 tetraploid background, which didn't seem to be as selected. The second was another lineage 2 donor in the Ho501 background. On the other end of the scale, probably the more interesting end, we have some tetraploid backgrounds in these synthetic derivatives, which were much more were selected at a higher frequency by breeders in the program. So it's interesting to see that the breeder selection at an earlier stage in the field is linking up with what's been predicted as being as good yielding. So obviously the breeder's eye is a real phenomenon. So looking at some independent evaluations. So these are the independent evaluation trials from the summer where we took the 25 highest yielding predictions, the 25 lowest, and the 25 most similar to the recurrent parent Robbergus. We did that because we want to be able to see if we can accurately distinguish the ones which are going to yield better than Robbergus. So that's why we took that third group. So we were still doing the data analysis on this, so I won't discuss the results today, but we can look at the predicted performance within the trial of those groups. So that's the 3 prediction groups that I just discussed in terms of grain yield, and this is a predicted value for Robbergus in purple. What we typically observe is a move away from what we call the adapted wheat when working with the synthetic derivatives. And what we learned from those histograms in the Meganam nursery is that to summarize the phenotype diversity in the primary synthetics are typically earlier flowering, they're taller, and they have better yellow rust resistance. So that's probably due to the rust resistance gene we mapped. But in terms of the taller and the earlier flowering, that's not necessarily a desirable trait from the breeder point of view. And what we see is this move away from the robust baseline when we're crossing with the primary synthetics, as you can see here. Interestingly, the earliest flowering group was the ones which were meant to be the highest yielding. So we haven't quite worked out why that is yet, and that's something that we're currently exploring. But it's this disruption to local adaptation, which is a challenge when working with these diversity resources. And that's something that we have a PhD student just starting with us called Megan Green, who's exploring some of these questions in more detail. So this is a nice outcome of that work with CARIS. So KWS are involved with this, and it's a BBSRC funded project in collaboration with the University of Cambridge as well. So here we're looking at, through simulation, if we can simulate some populations, typically backcross populations, where we're capturing diversity from plant genetic resources, capturing as much diversity as possible whilst trying to maintain local adaptation at certain elite regions. So it's an interesting project there, and related to that last slide of challenges of working with plant genetic resources. We're also looking at combining ability and trying to improve the estimation of that when using plant genetic resources. That's more thinking about work from the hybrid side, then also trying to form a training set for predicting combining ability in terms of genomic selection for local adaptation in the elite gene pool. So trying to find elites which are very good at maintaining local adaptation when crossing with different plant genetic resources. So this is a kind of small side project I have going with Richard at the moment. So Richard's the one with all the crossing experience. We're trying to directly cross Agelot's Taoiseach with modern wheat, and that's ultimately because we're trying to find a cleaner and faster route to accessing the diversity reserves of the D genome donors. So we're looking at and also trying to reduce the disruption to the A and B genome, because if you make a direct cross between Agelot's Taoiseach and hexaploid wheat, you're only changing the D genome. So we're doing that using a line from the Agricultural Research Institute in Hungary, listed in this paper here. So it has two recessive crossability alleles, which is meant to allow for crosses between wheat and rice. We're testing it with Taoiseach here to see if it works. And you can see from this picture, we've had some positive results. It does seem to be setting seed. So we're trying to then back cross these crossability alleles into more locally adapted elites as well, and develop some assays for marker-assisted selection too. So in the next couple of slides, I've listed some of sort of my highlights or NIAB highlights for going forward with this diversity-enriched material and what we're using it for sort of newer projects and projects in the future. We're always interested in phenotyping in different environments and under different conditions. And this is a collaboration with ICRISAT in Hyderabad, where we're looking at screening our NAM populations through a high throughput phenotyping technique, which is their LESI-scan gantry system, under a controlled environment, and then also their heat season as well. So we'll hopefully get some interesting differences in terms of performance or growth characteristics under heat stress. You can see in this trial, if you look carefully, that there are some lines which seem to be doing quite well. So that's an interesting, it'll be an interesting project. The trials are now completed, and we just got the data to analyze. So we have a University of Cambridge master's student who's starting with us this month to work through this. And we're hoping to do some QTL mapping for performance under heat stress as well. We have some international interdisciplinary projects starting as well, and these are really exciting projects using our diversity resources. So the one that I'm showing here is called the Wheat Alliance. So we're looking at boosting sustainable wheat nutrition through microbes. This is funded by the Nordis Foundation, and it's led by Professor Simona Raduțiu at the Aarhus University. There's a lot of different partners involved, as you can see listed there, and the project's just started, so it's all fairly new. We're looking at harnessing the potential of plant microbe interactions, so using a wider genetic diversity pool or a wider gene pool by using our diversity-enriched wheat to try and identify lines which have a beneficial root microbiome, with the ultimate aim of trying to then map genes related to that, map the genetic space that's involved in that, and then try and improve nitrogen and phosphorus uptake in wheat ultimately, using that relationship. So it's an exciting project there, and again, another opportunity for us to explore some of the genetics in these populations. We have some great PhD students as well. So these are two projects here which are using the diversity resources. So Anissa's project is looking at NIAB synthetic wheat, which have really good septoria resistance. So that's quite common when we're looking at the primary synthetics. And then she's mapping these in the downstream populations with the aim of identifying the genetic control for that, and then trying to do some fine mapping for genes coming from the D genome background, like we did for yellow rust. On the brown rust side, we have a new PhD student starting with us called in January, who was a master student with us last year, called Wright. So Wright is looking at tetraploid wheat, and we find that in this case, a tetraploid wheat is very resistant to has a lot of resistance to brown rust. You can see some clean wheat there. And Bright's looking to explore the genetic control of that and see if that tetraploid genetics is effective in the hexaploid wheat background. We also have a project ongoing with EBI, led by Sarah Dyer at EBI, and here we're looking at using open source genomic tools, making open source an open source genomic tool where we're using some of our populations that I've spoken about to upload to the ensemble plants, which is a genome browser run by EBI. There we will have quality controlled sequence data for those populations. So the idea is that users can order the seed, upload their own phenotype data for those populations, and then be put through a kind of rigorous association mapping pipeline to explore basically QTL mapping or association mapping within a browsable genome context. So they can really explore, the genetic regions in detail behind their QTL hits. So that's 7 crop plant species are involved in this project. So it's quite ambitious with several mapping populations for each species. So it's definitely something to keep an eye on. And then this is my last slide. So thinking about germplasm availability. So a lot of the material here has been funded by the BBSRC, and we're always trying to make this available as possible. It's challenging because there's so much of it. So we're currently working in collaboration with the GRU at the John Innes Centre to make this material available via so you're able to order it via Seed Store. So we've done that with a lot of the collections already, including the primary synthetics, and we're hoping to get the NIAB winter wheat synthetic NAM population fully uploaded, so over 3,000 genotypes, by next May. So it's an ongoing effort there. The population's genetic marker data, phenotype data, is available on the NIAB GitHub. If you want to read some more about some of the genetic resources and the availability of data from the populations I've spoken about, the NIAB website's the best place to have a look at. And then just to say that we're always looking for new partners in different wheat growing regions for testing some of this diversity resources or intercrossing it into some of their own elite wheats. So please get in touch with us if you're interested in collaboration, or even if you are ordering some of the material via Seed Store, you can still touch base with us and we can give some advice from our experience with the material as well. So I have attempted here to thank everyone who was involved in those projects that I've listed and the different partner institutes. That's definitely challenging, and I'm sure I've missed some people or an institute, so I'm really sorry if that is the case. And here I just want to acknowledge BBSRC, who funded a lot of this work via PhDs or larger consortiums with multiple institutes involved, like the ones listed here. And also just to acknowledge my NIAB colleagues and say thank you to them for all their efforts over the last couple of years, because the work I've discussed is a large collaborative effort, and it's been wonderful working with them. So thank you very much.
Okay, thank you very much. Yeah, thank you very much. It was a very interesting talk on genetic resources you're developing. I believe there are a couple of questions. I'll start here. The first by Clément de Debutant. By using Robigus, which has dicoxoides in its genetic background, do you think it could bring a bias in the recombination events that occur in your population coming from a cross with a tetraploid, such as between dicoxoides and dicoxum?
Yeah, that's an interesting question, and I suppose the tetraploid NAM is the one that we've tetraploid winter NAM is actually the smallest NAM population out of all of them, and the one that we haven't explored as much detail. We did find Robagus was one of the better elites for crossing into, so it generally had a good combining success with some of these diversity resources. So it's interesting thinking about it from that angle. I know it does have that large integration from Triticum dicoides. So whether that's one of the reasons, we haven't been able to narrow down on that. But Robagus is one of the Magic Elite founders as well. So that's one of the lines we have a chromosome based assembly for. So it's something that we could potentially explore going forward. It's definitely an interesting question. I do note, just thinking about the numbers, that the Robagus winter NAM is the smallest NAM population. So whether that had a higher failure rate, that might be true. Whether it's due to that, I couldn't say. But yeah, it's definitely an interesting point.
But to your knowledge, you haven't seen any reduced recombination in the populations that you've looked at so far?
That one, I don't think we have currently genotyped. So if I think back to the NAM populations, there's two which we have genotype data for, and that was the one that we haven't yet sequenced or genotyped. So I can't say about the recombination levels yet, but hopefully that's something that we can look at in the future.
Okay. Here's our next question by Uzalitan, if I pronounce that right. And he actually has two questions. Question one, in grain yield slide, you showed blue result. Is there any specific reason to use blue instead of blup?
Yeah, that's a good question. So we used the blue there as it's typically based on a trial design. So we've removed the blocking effects, which is why it's a blue rather than just an observed mean. So that's why we've used a mixed linear model to estimate the genotypic effects. The reason why we use blues, they don't have any sort of shrinkage towards the mean, which blups would do. So when using two stages of analysis, such as what we do typically for an association mapping pipeline or genomic selection, there's normally a random factor such as kinship used in the second stage of analysis. So if we use blups followed by blups, I think that's two levels of shrinkage, which can reduce the variation quite a lot. So that's probably why we didn't use blues, and sometimes it's easier to work with blues in that scenario. If we had, if we just wanted to look at the variation from stage one, or we wanted to explore so if we wanted to explore data where there was a lot of varied replication, then I think we'd lean more heavily on using blups.
Okay. And the second question is, how did the training population, or how training population genetic differences affect in prediction accuracy, as training population is diverse in genetics while test population is sister line in most cases?
I kind of see it as the other way around. I mean, the fact that the training population for that experiment was selected by a breeder, I'm more worried that it's narrowed the diversity, whereas a test population contains 15 nested populations, which include lines that haven't had any selection. So it's the whole of the population. So my only concern would be that the training population has reduced variation compared to the test population, and maybe that's where the question. What the question meant. It depends what the breeder selected from. So I mean, Carrison in this case was the breeder, and I don't think he always selected necessarily on grain yield as much earlier in the season, although we did see that the breeder's selection was linked to grain yield. But it seems that the prediction accuracy is good enough, and some of the variation that we observed in the field suggests that it has worked in terms of the test population so far, even when just weighing out the seeds for what was going in the yield trial, some of the lowest prediction ones, I couldn't find enough seed to actually test in the packet. So hopefully that's a good sign that it has still worked, and we'll get some positive results from that.
Okay, thank you. All right, next question. We are working in fine phenotyping using non-invasive sensors for drought and heat. In Nanaji Plant Phenomics Center, New Delhi, India, possibility of NAM phenotyping. I guess, yeah. So maybe this is a idea for collaboration, I guess.
Yeah, please drop me an email. I appreciate that. Thank you.
Okay, great. Currently our last question. Thank you for the impressive talk. What is known about the stability of genomes and synthetic derivatives?
Yeah, that is an interesting question. On behalf of the NIAB, the NIAB synthetics that we work with, they tend to be quite stable. When we genotype multiple replicates of the same line from different sources, it does tend to be quite stable. We find actually, thinking back to some of the work with the tetraploids versus the hexaploids, what we call our hex populations, that's where we form crosses between the tetraploid and the hexaploid. Some of those individuals are the ones that actually seem less stable. So in some cases, we find one that's missing a chromosome, and I think it's due to the tetraploid, the hexaploid ploidy. Sometimes it's worked out by backcrossing, and sometimes it messes things up. But in terms of the primary synthetics, speaking on behalf of the NIAB ones, they do seem fairly stable. I have read that they can be unstable, and they do seem more prone to outcrossing as well. So that's interesting, thinking about the ear morphology. So it's something that we're always having to keep on top of, and something that we're trying to do for the new projects is make sure we're maintaining these genotypes going forward, because it is always a challenge to keep something as it should be if it's prone to outcrossing in a field of lots of stuff going on.
Yeah, it's very interesting. Okay. All right. I think this might be the last question. Has this diversity been explored for stem rust resistance, especially against UG99 race? If not, are there any plans to screen the diversity panel for this purpose?
Yeah, I think there's a lot of work still to do in that space. So I can only speak on behalf of what we've done in terms of the Designing Future Wheat project. And obviously we're looking at septoria and brown rust, but we screened for yellow rust resistance in the Designing Future Wheat project, and that was just using natural infection. So two of the trials were untreated. And that was 2017, 2018. And then we had another trial in 2021, which was treated, but again, just scoring for natural infection. So we don't really know if which isolates we're necessarily working with. It was just based on the natural environment. So going forward, that's definitely something that we want to characterize in more detail.
Great. Okay. I guess that's the end of the questions. I guess I had a question about the various D genomes, whether there's any kind of relationship between which D genomes may have functioned or not functioned relative to the particular genome that hexaploid wheat acquired 10,000 years ago, and whether there's something significant about that original hybridization that's not captured in other hybridizations.
Yeah, I think that's interesting. So the lineage 2, which is where the original D genome donor comes from, I find it interesting that it's easier to form primary synthetics when working with that lineage. Again, I can't say why, but it is, and tended to be what was more selected by breeders. So whether that's an indication of that side of things, I'm not sure. And yeah, it's definitely an interesting question.
Cool. It's very curious. Oh, someone else just added something. Sansaloni et al. 2020 had represented 80,000 wheat accessions for trait selection. Is it required to create synthetics and magics and NAMs?
Yes, well, that's an interesting question. I think it boils down to the difference between association mapping and QTL mapping, maybe. In association mapping, you might have a single rare haplotype in that very big collection there that isn't represented very well, and you'd never have enough statistical power to associate a a trait to it. But if you use a multi-founder population, you are replicating that haplotype more frequently throughout the population, so you have more chance of associating an effect with it. So yeah, I guess that would be my argument there.
Okay, there are no more open questions. I really want to thank Tally for an amazing presentation describing just incredible amounts of work to generate all of these materials. And I'm sure there's going to be some great science emerging from all of these materials in the future. And so once again, I thank Tally Wright. And here's another someone else, Vijay Raja Minakam also said amazing presentation. So I want to thank both you and all of our attendees for joining us today for this webinar. And once again, if you should expect to see this recording arise in YouTube in case you didn't get it. Here's another comment, brilliant work by Bright Flaque. So anyway, and we look forward to our next webinar, which will happen next year, either in January or February. So stay tuned for that. Thank you all again, and we'll see you next time. I appreciate it. Thank you so much.
Thank you, Josh.